Nearly every medical intervention humans have ever developed shares one basic property. Its effects stay contained to the person treated. A surgery, a medication, a vaccine, whatever changes they produce live and die with the patient who received them. Human germline genome editing breaks that historical pattern. If an edit is made to an egg, a sperm cell, or a very early embryo, the biological mechanism dictates that the modification does not stay contained to a single body. Because the technology alters the earliest cells in development, that specific edit is permanently copied into every cell produced by every subsequent cell division for the rest of that person's life. Crucially, that includes their reproductive cells. The edit is passed automatically to their descendants, generation after generation, with no current technological way to extract it once it has entered a lineage. Every other edit medicine makes stays with one patient. This is the one kind that doesn't. Popular coverage of gene editing frequently blurs different technologies together. To understand the boundary we are dealing with, we have to draw a strict line between two distinct categories. The first category is somatic gene editing. This modifies specific tissues within an already living person's body. This split-screen diagram illustrates the distinction. On the left, we see how somatic editing works in approved CRISPR-based therapies for conditions like sickle cell disease. Doctors modify a patient's bone marrow cells, and the edits remain strictly confined to that specific tissue. While those changes drastically improve the individual's quality of life, they fade out completely when the patient passes away. They are never transmitted to any children that patient later has. On the right, we see the mechanics of germline editing. Because this method targets the earliest reproductive cells, the edit at the root node is automatically populated down into every single subsequent branch of the family tree. That automatic, unstoppable transmission places germline editing into a completely different tier of consequence than any gene therapy currently approved for clinical use. Somatic editing is a medical treatment. A germline edit is an engineered inheritance placed into a family line before the person carrying it or any of their descendants can consent to the change. In November 2018, Chinese scientist He Jiankui announced that his research team had edited human embryos and that those embryos had resulted in live births. The team used the CRISPR-Cas9 system to alter a specific gene called CCR5, aiming to give the resulting children genetic resistance to HIV infection. This visualization shows the biological rationale behind the target. A naturally occurring variant of the CCR5 gene already prevents HIV from entering immune cells in people who carry it, giving his team a scientifically plausible mechanism to replicate. But a plausible scientific rationale is not the same as a medical necessity. Highly effective, non-editing methods already exist to prevent an HIV-positive parent from transmitting the virus to a child. The announcement triggered immediate, near-universal condemnation from the global scientific community, including from scientific institutions within China. Critics focused on multiple specific failures in how the experiment was run: an inadequate informed consent process, a severe lack of transparency, and flagrant disregard for established guidelines. The legal response was equally definitive. In 2019, He Jiankui and 2 collaborators were convicted under Chinese law of illegal medical practice. He was sentenced to 3 years in prison and permanently dismissed from his university position. The 2018 experiment now serves as the field's defining cautionary tale: the deployment of a permanent, hereditary edit for a problem that standard, untargeted medicine could already solve safely. The popular reaction to the 2018 scandal assumes that the scientific community completely and permanently banned the creation of edited embryos. The actual documented statements from the field's most authoritative bodies establish a much more conditional position. In the immediate aftermath, the organizing committee of the Second International Summit on Human Genome Editing convened in Hong Kong to issue an official response. Their exact conclusion stated that clinical trials remained too risky at this time, but that ongoing progress made it necessary to define a rigorous translational pathway towards such trials in the future. Before we look at the exact line scientists drew, take a second to subscribe to Your Dose of Wowness. This is day 149. The 9th episode of our 10-part arc on boundaries, and you won't want to miss tomorrow's finale. Separately, a group of prominent genome editing scientists published a call in the journal Nature for a global moratorium, backed by CRISPR technology pioneer Emmanuel Charpentier. Leading geneticist Feng Zhang also lent his authority to this specific call for a halt. This diagram maps out the crucial distinction these scientists drew. They called for a strict pause on clinical applications, meaning no creating pregnancies, but they explicitly supported continued fundamental research into germline editing itself. The pressure to eventually cross that line into clinical use remains active. Russian scientist Denis Rebrikov later announced his own intentions to pursue germline editing procedures, adjusting his targets as he sought regulatory approval. The actual global consensus among researchers isn't never. It is a deeply unresolved, not now, and not like this. The threshold remains closed today primarily because of severe, unsolved technical risks. The foremost concern is off-target effects, unintended genetic mutations occurring elsewhere in the DNA sequence, which would then be permanently passed to all descendants. A secondary technical risk is Mosaicism, where an embryo develops with a dangerous, unpredictable mix of successfully edited cells and unedited cells. Beyond the technical hurdles sits an unresolved policy gap. Practically no international consensus exists on where a regulatory line should sit between editing to prevent serious heritable disease and editing for elective genetic enhancement. And beneath that is an unprecedented bioethics problem. Medicine currently has no framework for obtaining consent from people who do not yet exist for a permanent biological change they cannot undo. This mirrors the tension we covered in Day 139 regarding cryodynamics. We are looking at actively contested science, where experts examining the exact same data draw deeply different conclusions about how to proceed. But germline editing functions differently than the other engineered boundaries we've covered in this arc, like Day 144's Rubicon or Day 147's V1 speed. This diagram shows the distinction. Previous thresholds were calculated, solid lines built by institutions after a danger was already well understood. The germline threshold has no single agreed-upon metric. It is a shifting, blurred gradient being actively negotiated in real time. Human germline editing remains the exact intersection of extraordinary therapeutic potential and a scale of biological irreversibility we have never had to reckon with before. Tomorrow we close the arc, Day 150, Building in a Way Back, looking at the real systems engineers and institutions have built specifically to catch a decision before it crosses a line, like the ones we've covered all 10 days.
Thank you for sharing this moment of pure wonder. You are the true magic that fuels this journey. Until next time, remember to shine bright.